Hi, my name is Simone Bianco. I am principal investigator and director of computational biology for Altos Labs. And I'm very sorry I cannot be there in person. For personal reasons, I cannot travel, but I'm very happy to give you this contribution in this video form. And thankful to the organizers and the National Academies and the African Academy of Science for the invitation. Today, I'm going to give you a perspective on the, how artificial intelligence can be used for design of biological systems. And especially, I want to give you some ideas of the field of synthetic biology, how computers can really help the field of synthetic biology. First of all, I am from Altos Labs, which is a new life science company that works on restoring cellular health and resilience to cellular rejuvenation programming. So you're probably familiar with the work of Shinya Yamanaka. He won the Nobel Prize in Medicine in 2012 for his work on inducing primary stem cells to chemical factors, now called the Yamanaka factors. Taking any cell, a cell of any kind, and using these 4 factors to making it revert to its pluripotent state, and this is called cellular reprogramming. Altos works on partial reprogramming, which is a way to reprogram cells that doesn't make the cell lose its identity. So if we start from a somatic cell of any kind, then for example a muscle cell, the cell will stay a muscle cell, and this is done to in several ways. So my group at Altos is called the Computer-Aided Rejuvenation Group, and it has a very diverse set of people with a very diverse set of skills, as you can see, from the atomistic molecular dynamics modeling to hybrid AI, to biochemical networks, epigenetic modeling, modeling the immune system, and AI foundations. The aim of my group is to build predictive mathematical models and computational methods to increase our understanding of the mechanisms of cellular reprogramming and rejuvenation. But it has an edge with, let's say, any other computational biology group, which is that we want to establish design principles of rejuvenated cells and tissues. And this would come as part of my perspective for AI in biology in a second. So what is it that we do? Well, so we work specifically on synthetic biology. Now, synthetic biology is a branch of biology which aims at taking what biology has built and modifying it or tweaking it or using modules from biology to make biological systems that have never been realized by evolution. As such, really, if you think about it, is a way for biologists, or in this case, synthetic biologists, to really implement the design, build, learn, test circle that is very dear to engineering. So it's really bringing an engineering to the field of biology. Now, the problem with synthetic biology is the design of biological systems is undermined by the fact that there is an extreme complexity in biology and intrinsic stochasticity. I'm going to go to that in a second. But what we've been able to do so far in the field is really akin to this analogy of the blind man and the elephant. We can really look at only a specific part of the of a biological system, and we've been able to understand them, but we've not been able to get to the full picture. Just similar to what you're seeing on the screen, all this blind man and the elephant are capable of looking at a small part of the elephant without being able to make it all. And only by bridging all the knowledge, we can get to the full picture. And so you may wonder, is the computational design of not only a Junior cell, but any cell really possible? And so I'm going to give you my perspective. So first of all, as I was saying before, biology is complex. There are, in our understanding of any biological processes, there are unique knowledge gaps. For example, even some of the most studied pathways, like the p53 pathway is not completely understood. Not just that, but also the dynamic at the cell level is at multiple scale, both in terms of space, from the atom of the molecule to the whole cell, to even tissues and the whole organism too, but also in terms of time. So some processes happen very quickly, other processes like diffusion happen very slowly. And so understanding this complexity is really challenging, and it is, in my opinion, a requirement to really do effective computational design of all cells. Also, cells during evolution, they have developed very exquisite self-organization principles. For example, we know that organelles like the nucleus are put there by evolution to keep chemical reactions separated, and this is again many times poorly understood. For example, how many organelles do we have is something that is tuned by evolution, and it's a self-organization principle. But also things like the ratio between the size of the organelle and the size of the cell, and so forth. Something that is very dear to me is the fact that as a physicist by training, is that noise really in biology is not a bug, it's a feature. But it is not really treated as such. And so if you think about other disciplines like physics, there has been there is a long literature, I want to argue 150 years of work in statistical physics, for example, starting from Gauss to information theories, going to Boltzmann, to understand the role of noise in physical systems. If you think about engineering, there's control engineering that deals with noise in engineered system. The same is not true with biology. And so my opinion is that in order to understand the process the processes that underlie biological assembly that will lead us to synthetic biology, we really need to understand the role of noise. And of course, evolution, I don't even need to mention evolution as a really a factor in our design. So if you take all this together, what this means is that there are really very few rigorous design principles or general strategies beyond the trial and error for cellular design. However, if we do consider subsets of the cell, we can consider cells and their biochemical networks as dynamical systems. We can assume that, for example, cell state is a point in a multidimensional space where each feature, for example, the shape of the cell, its rate of production of a specific protein, and so forth, is a component of the vector. As such, the cell is a point in a multidimensional space that in time evolves from one steady state to another. And so there are many time and space scales, as I was saying, and there are many variables. And there is actually a formalism for treating the cell as a dynamical system, and that is systems biology. And you're probably there's no you wouldn't be surprised, of course, to know as you all do, that systems and synthetic biology are almost always part of the same sentence. How would you go about designing a biological system using systems biology? Well, you will take your for example, your network of or your understanding of your network, biochemical network of interest. You would simulate it with a number of parameters, and then if one is interested, for example, in looking at the system in a where feature 1 is low and feature 2 is high, one can extract design parameters. Now, the problem with this approach is that many times our understanding of the biology is limited, as I was saying, and also our models are coarse-grained. They assume that these networks live in isolation, which is never true, and this many times leads to these parameters being not close to the experimental. And so this is of course, it's a flawed way, although it has been, of course, very successful. Now, in the last decade, a new strategy for design design of biological system has evolved, and it is this data-driven design strategy where you collect a bunch of data, extract features to establish a phase space from which you can learn a model. And many times this kind of system, this kind of algorithm is a neural network of some form, and using this algorithm, one can predict optimal design choices. And so what this brings us, so joining these two parts, this data-driven and model-driven, brings us closer to our understanding of synthetic biology as a computational science with commas. And so this has led us to a number of advancements. For example, we have developed a method to assess biophysical quantities of engineering molecules, which we just published. We have developed with John Duber at UC Berkeley a model-driven design of the yeast peroxisome for penicillin biosynthesis. We have developed and we patented this a screening algorithm for synthetic genomes. We have developed algorithms to screen combinatorially input-output relationships from only a few experimental measurements, and this is something that we filed. We have developed algorithms to design chimeric antigen receptor T cells. This was just accepted on Science. We have developed algorithms to infer a genotype from a receptor phenotype. And just lately, since we started in Altos, we have developed models to understand chromatin rearrangement during reprogramming. And so this means that, in my opinion and in our experience, bridging artificial intelligence or data-driven design strategies with model-driven design strategies will give us principles for larger and larger systems. And we hope that in the next decade we can get to the point of really designing a synthetic cell. I'm going to give you now an example of our work on the AI-driven design of chimeric antigen receptor T cells. This was work that was started when I was at IBM Research, and it's continuing now that I'm in Altos, and it's led by a wonderful researcher, Shangying Wang, and it is with UC San Francisco. So you're all familiar, I'm sure, with chimeric antigen receptor T cells. This is a wonderful strategy for cancer therapy based on the re-engineering of the immune system, where a T cell is extracted from a patient and then re-engineered so that it is functionalized to selectively attack cancer cell or increase proliferation or its stemness potential. And so there have been at least 3 generations of chimeric antigen receptor T cells, and we call them CARs from now. The first one uses an intracellular module that is functional with a synthetic external receptor. This has been shown to never work. The second generation uses a co-stimulatory domain from either CD28 or 41BB genes to increase the phenotype of the T cell. And the third generation, which is the one that I'm going to show you, is capable of using 3 motifs to have a combinatorial efficacy in phenotypes. So the work that we do is basically the following. Imagine that you can purify functional motifs from specific genes that are known to have specific properties. For example, increase proliferation of T cells. This is basically looking at using a language analogy of a native sentence from which we extract words. Now, what we want to do in using synthetic biology is really to re-engineer, so to build new sentences with new meanings, where the meaning is the phenotype, by combining different words. And we can do it in 3 positions with a tail. And so, sorry, let me go back. So this is, of course, if we want to do it experimentally, is extremely expensive. And so we use smart algorithms to do this kind of, to extend this library to do this kind of work. And we want to get to the point where the algorithm can suggest specific design principles for cells with cytotoxicity, increased cytotoxicity, increased stemness, or any phenotype for that matters. This is just a schematic representation of what we do. We use a smaller set of our library. Our library is composed of 13 motifs, which makes up to over 2,300 combinations. We use about 10 of them, and we use this randomly chosen training set to train a neural network after triplets of CAR-T motifs have been encoded using one-hot encoding. What this is doing is really recasting this linear sequence into an image that then our convolutional neural network can understand. And the output is the prediction of the phenotype. For example, increasing proliferation or killing capability of the specific phenotype. So these are our results. On the validation point, we can get high accuracy for phenotype of cytotoxicity, which is related to killing cancer cell, and stemness, which is related to the establishment of memory of the T cells. And so, using this this trained algorithm, we can generate the whole library synthetically, and then select those triplets, for example, here, that have high cytotoxicity and high stemness. And so this really goes towards this idea of data-driven design principle. And then we have gone to the lab and validated that these are actually constructs that have increased phenotype. This gives us some confidence that this design strategy is optimal for this work. Not only that, but something that is very interesting is that the algorithm built in this way is capable of getting a higher level biological features from the data. For example, if we use it to predict what will be the change in phenotype if given a certain set of domains, we add a third one of a specific kind without giving it its exact sequence, but only the class it comes from. For example, 41BB or CD28, the algorithm is capable of telling us the result, which we then can go back to the biology and verify using biochemical networks. And so again, if you want more information, this is on the bioRxiv and it's forthcoming on Science. So let me go to the conclusion. So what is the role of AI in the life science? Well, going directly from the idea of this data-driven, right? So that using data to establish design principles, we can really delineate at least three important parts that constitute, in my opinion, the role of AI in the life science. The first is classify, and this is probably what you're most familiar with. So we have two kinds of cells. One is red, the other is blue. We train an algorithm to recognize red and blue, red from blue, and then we have a new cell. We ask, is it type A? Is it red or blue? And lots and lots of problems can be recast as classification problem. For example, segmentation, image segmentation is a classification problem. Is this pixel inside the boundary or outside the cell? The second is AI as an accelerator. So especially when we bridge mechanistic modeling and when we try to do design to mechanistic modeling, we may find ourselves in a situation where the phase space of all potential parameters, as I was showing at the very beginning with the model-driven design method, is enormous. You know, a set of 5 equations can have 10 parameters, which may take years, or hundreds of years, even thousands of years, as my postdoc Shengying used to tell me, to be explored fully. And so we can use artificial intelligence to learn the mechanism-based model and use it to predict the dynamic for many parameter sets for a very in a very small time. And this was published by my postdoc Shengying with Lingchong Yu a few years back. And the last but not least, and I think this is where there has been less work, but more, I think where it's more exciting, is where AI is a designer. And it's similar to the work that I was showing before on chimeric antigen receptor T cells. I think artificial intelligence has the potential to derive the discovery and the design of new biological systems, not only through the simpler, in some sense, work that I've shown you before, using that structure, the network structure, but to more refined and exciting paradigms like reinforcement learning, generative models, autoencoders, and using principles of explainable AI. So I want to conclude by acknowledging my group at Altos and my former group at IBM, as well as collaborators at UC San Francisco in the Wendelin group, and the National Science Foundation for sponsoring part of this work. Thank you all for the attention, and I look forward to the discussion.